To welcome Perihan Nalbant here, and she's going to talk about spatiotemporal regulation cytoskeleton dynamics. I guess looking forward to the talk. Yeah.
Yeah, thank you very much for having me here as well. A bit late, but well, I can't help it. The airline didn't want to bring us here. So anyway, so I was initially scheduled last week with all those cell biologists who did already talk. But this week is also very nice. So then I'm kind of embedded in a bit more like mathematical environment. So, but nevertheless, it's going to be cell biology today, and I will talk about. The title says spatiotemporal regulation of cytoskeletal dynamics. In particular, I will talk about the actin cytoskeleton. For me, naturally. So we will have several parts, and I didn't put up a sort of what do you call it? Like topics, right? So I didn't want to introduce that because I try to kind of try to be it like a story. So I hope you don't mind not knowing what comes next. So all right, the actin cytoskeleton we know, and you probably heard last week more than enough, is very important for shape definition and cell behavior, obviously. So despite actually that the biochemistry of actin polymerization is quite the same in different cell types, we do see much like big differences in the overall cell shape in different cell types, as we see here, like bone cells, which look very much more rigid and they don't look very dynamic in contrast to neuronal cells. They look much more dynamic, thin, and much more plastic or show more plasticity. And also similarly, those migrating cells, here I put up as example the fibroblasts. They have a very defined shape, polarized shape, we say, with the front and the back part, in contrast to other static cells like the epithelial cells, although we will see that they are not very static as it looks like. So all those different shapes most often are associated with those distinct functions, obviously. So let's look at the migratory cells. We all know how migratory cells look, but it is really always nice to see also at the same time how the cytoskeleton looks like. For me at least it's always very nice to look at. But it is correlated to the function that there has to be a leading edge during migration, and there has to be a back which is basically pulled afterwards. And this is mirrored actually in the composition of or organization of the cytoskeleton that we have here in the front, other actin structures as compared to the back. So it looks very different what we have in epithelial cells. They don't look very polarized, at least not in 2 dimensions. In 3 dimensions, 3D, you do have an apical and basolateral polarity. But if you look from top to those cells, you will see the actin cytoskeleton basically surrounding like a belt the entire cell. And the main role here of the actin cytoskeleton or this actin belt is basically to keep the cells together, as it is also important for the integrity of cell-cell junctions. So the morphology of the cells or the change of morphology is not only important during the cellular behavior as it adapts once after the division, cell division, for example. It is also important for the adaptation of new cell behaviors, as we see here, for example, during the epithelial to mesenchymal transition during embryogenesis, for example. As a nice model system, we do use often those MDCK cells. Those are kidney epithelial cells from dogs, actually, and we stimulate those with hepatocyte growth factors. And there we see really nicely, so those cell colonies, if you don't treat them at all, just leave them in culture, they will turn around, they will move a little bit, but they will stick within the colony. However, if you add this growth factor, they will change their morphology completely and adopt the migratory behavior.
What kind of cell are these?
Excuse me?
What kind of cell are these?
Those are MGCK cells, classical kidney cells from dogs. I don't know yet now this what the M is for. After a while, yes, they do. I mean, they don't really start at a certain point, but it is not a synchronized system, I would say. I mean, they are like cell divisions you do see here also at some point, but on later stages. So this dramatic change in morphology is actually also mirrored in the dramatic alteration of the actin cytoskeleton as well. So this colony is also, we do also see the F-actin cytoskeleton here. We focus this on the most ventral part where the cell substrate adhesions are. And that's why, to see this actin belt surrounding the entire colony, if we would have focused a little bit higher, you would again see basically the cell-cell junctions much more nicer. And then obviously after hepatocyte growth factor stimulation, we do have here completely different structures. All of a sudden some protrusive structures, some linear fibers, which I will explain later what they are. So this dramatic change is really also important to adapt a different cell behavior. So how are those different shapes established? I will explain basically two or three most important structures in cells that define the overall the shape and the dynamics of cell behavior. And first of it, we do have those protrusions, as I mentioned before. This is again the actin cytoskeleton. We have here a very narrow band of actin structures that look very much like a little bit fuzzier than the rest of it. Then we have those fibers, but those first front region is basically based on this, or it's called lamellipodium. I'm sure you heard about it last week. Who didn't? Well, so this lamellipodium consists of an actin meshwork, which is which is called quasi-polar. We know that, as you probably know, the actin filaments, when they polymerize, they have a growing end, barbed end. We call it plus end, and they have a pointed end where the polymerization starts. So, but overall, you don't have like linear fibers, so you can obviously just point to one fiber with a minus end or plus end, but overall you will have an enrichment here in the most edge or very edge of the cell, more plus ends, and here a little bit further inside more minus ends. So this means that the polymerization, which happens here more in the front of the cell, will build up some force and push the membrane outward. So we have overall a protrusion generated. So those processes which happen in this network or meshwork to generate this meshwork, they have been obviously summarized already over 14 years ago now. I can't even believe it. By Pollard et al. So this model is also called dendritic nucleation model, and describes basically how all these different regulators work together to initiate polymerization, to pursue elongation, then recover the actin subunits, and then to basically ensure the cycle. So altogether works in a continuous mode to ensure persistent dynamics of the leading edge. So the second type of structure I would like to explain are those contractile fibers, or those linear fibers I first described or pointed out. They are actually actomyosin fibers. We have an enrichment in the back, but also some in the front. So the way how it works is basically that we have a myosin dimers that are anti-parallelly organized, and they both bind an actin filament, and those are actin motors, actually motor proteins, and both of them now are trying to move towards the plus end, but because they are attached to each other, they can't go anywhere, right? So the consequence is that they pull the two anti-parallel actin filaments towards each other. This is how contractility actually is generated, like this. It took me an hour to do this, so all right. But so how do those stress fibers or those anti-parallel filaments that are now just pulled together, how do they define shape, cell shape? Because if you have like a rubber band here without any anchor points, you won't have any tension, and you won't be able to define any structure, yeah, or any cell morphology. So the answer is obviously anchor points, which stress fibers are attached to, as we see here in this migrating cells you saw before. We have here at the end of the stress fibers, at both ends here in this cell, those accumulations of adhesion proteins that are called integrins, and we call those structures focal adhesions. So I will go a little bit more in depth into this structure, because the two together, stress fibers together with these adhesion elements, the focal adhesions, those are basically mechanosensory elements that sense forces basically from the outside and transduce those forces inside the cell. We call it outside-in signaling. So how do those structures now sense force or different forces from the outside? The first elements that have to be activated by the extracellular matrix are integrins. Those are receptors for extracellular matrix proteins, and they are activated, and they transduce basically those activity inside the cell and bind or activate a protein called taline that is force sensitive, and it is stretched on different strength. So the more force is applied from the outside, the more taline will stretch and open up binding sites for other more and more interaction proteins. So, for example, vinculin, which is then a signaling protein that interacts with the actin cytoskeleton or initiates the actin polymerization by recruiting actin regulatory proteins. Additional factors that are activated by this activation process are kinases, such as focal adhesion kinase, that can really induce several different pathways, even activation of MAP kinases and Rho GTPase activities. So I'll talk about Rho GTPases in a bit. And what is really important, I think, in this interaction between stress fibers and focal adhesions is that focal adhesions themselves are very dynamic. Yeah, so they undergo a turnover, such as they are getting induced, they mature, and they dissolve. So here I have a movie with EGFP paxillin, which is a member of those focal adhesion protein family or proteins, and we do see that some of these adhesions at the cell edge are dissolving here, and some are generated new. So why do they do that? Why do we need those constant turnover? Very simple. If a cell needs to or is required to move, it needs to, of course, lift its adhesions, right? So you can't move if you're stuck anywhere. So this is the very most obvious reason why we have this turnover. But in addition, also this turnover has been shown to be important for this force transduction as well. So details are still, I guess, under investigations. It is very complicated, but just to be able to undergo turnover makes the focal adhesions also more mechanosensory. Anyways, so all this is of course important also on a global in a global way, such as that basically this outside-in signaling, like those matrix properties or forces that the cell can sense, define now the shape of the cell. So the more rigid basically the matrix is, the more rigid do cells also look. So what does rigid means? They have more focal adhesions, they have more stress fibers, they can basically stand those forces by reacting or by generating those really rigid structures as well. So in addition to that, the extracellular matrix is a really key factor for cellular differentiation. Yeah. So we do see here. Sorry, let me go back again. In the brain, for example, we have very, very soft tissue, whereas, for example, in bones, we have more rigid matrix, extracellular matrix, and so the cells have to somehow adapt to those environments, environmental conditions, by differentiating in those cell types that are required in those regions, right? So, for example, Engler et al. Showed that mesenchymal stem cells differentiate or express specific differentiation markers depending on what type of matrix elasticity they have been are cultured on. So on softer matrix or on less rigid matrix, neuronal markers were more upregulated, whereas on very rigid matrix we have more markers or markers upregulated that are typical for bone cells. So this is was quite interesting. So now we have the cell, and it has those signals from the outside, and it is changing its morphology or is establishing its morphology. So how are those structures now organized in space and time? Yeah. So let me go back to the contractility and the localization in the cell. Okay. So we do see here that stress fibers are basically everywhere throughout the cell, organized. However, they are not really homogeneously distributed in the cell. So we have here in the front some contractile fibers and also in the back, but in the back it appears at least that the contraction or the contractile fibers are much more. This is what we do see in many cell types, that we have basically gradients of protrusiveness versus contractility, and they go along quite well actually, but they are excluding somehow each other. Yeah, so we have in the front some contraction, but much more protrusion, whereas in the back we have still actin polymerization happening, but much more contractile stress fiber organization. So what are the key components, or what is the key component for these localized generation or organization of those structures? Obviously, we are interested in the question of how those regulators of those structures are organized in space and time, and if localized activities are critical for those final shape establishment, for example. So I'm sure you have heard about Rho GTPases last week, but we are really working quite intensely with those proteins, so this is our focus, so that's why I'm talking about those. And those proteins or members of Rho GTPases are important for the formation of different actin structures. They belong to the RAS superfamily, and many of them are known. However, still the best studies on the, of those are the three probably well-known, best-known, RhoA, RAC, and CDC42. So those three proteins do quite different things. Yeah. So we have first this RAC1, which is very important or generates those protrusions in the front. And we have seen this already, this actin meshwork, which is described by the dendritic nucleation model. And it is important for pushing basically the membrane. And CDC42 does also induce cell protrusions, however, different type of cell protrusions, those called philopodia or filopodia. They arise also from within the lamellipodium. They align not anti-parallel, but they are parallelly organized. And they are thought to be important to finding the direction of the cell. So this protein is important, for example, during chemotaxis, for the directionality of cells, that they find really the gradient of the chemoattractant, for example. So lastly, we have RhoA, and which generates contractility, and it was thought that RhoA was mostly active in the front, but we will see something else in a bit. Because we thought in the back the contractility is the highest, that's why Rho must be mostly active in the back. So those proteins act basically like molecular switches because they can exist in two different states, active and inactive, by simply binding to GTP versus GDP. The GTP bound form is the active GTPase, and the GDP bound form is the inactive GTPase. So they can switch between those states. So obviously the localized activity or spatiotemporal activity is regulated by at least two, I won't talk about the third one, two regulator types, guanine nucleotide exchange factors. They exchange the nucleotide from GDP to GTP, and those GTPase activating proteins, it's kind of misleading, the name. Those proteins activate the intrinsic GTPase activity of the GTPase, so the GTP hydrolyzes and the GTPase falls back into the inactive form. So those gaps and gaps are important. So, and only the active form can obviously interact with downstream effectors that do all those nice things with the actin cytoskeleton. For example, let's look at RAC. Once activated, RAC can interact with the so-called SCAR protein, which is a nucleating promoting factor. That is important for the polymerization or initiation of actin polymerization by ARP23. So it is basically, it has the first role that RAC has in the lamellipodium is activating the polymerization. Secondly, RAC activates the kinase PAC, which induces or which inhibits this protein, ADF cofilin, which is known to basically break down the cytoskeleton or the actin filaments from the back. So if it's inhibited, then we have a stabilization, overall stabilization of the filaments. So RAC has a dual role. First of all, it induces polymerization, and secondly, it stabilizes. Overall, it's kind of an amplified process. So what happens if we now induce or introduce into cells a mutant form of RAC, which cannot which cannot hydrolyze the GTP, so it cannot be inactivated. It's activated all the time and everywhere. It is independent of spatial and temporal regulation. So we have everywhere those nice lamellipodia arising, yeah. And one would naively maybe assume that now the cells would migrate faster, but of course it's exactly the opposite. The cell migration is inhibited if you have a mutant like this in cells, in any cell as far as I know, actually. So So why is this so? I wrote it here already, that the cell basically needs a localized protrusion in order to migrate properly into one direction. Otherwise, if the protrusions are everywhere, it will just try to go in any direction, and it will basically at the end it is stuck at the same place. Well, so this was RAC, and rho A is this contractility GTPase has also multiple downstream effector pathways to generate basically those its function, yeah. So to transduce its signaling. So first of all, it activates rho kinase, which then phosphorylates myosin light chain. That is then active and can really generate contractility of those anti-parallel filaments. Secondly, rho A is known also to activate so-called formins. Those are specific or distinct group of actin nucleators, and those formins are thought, or some of them are shown to really induce actin polymerization. So rho A basically generates filaments, right? Induces the filament generation and their contractility. It's basically both sides are active. Both functions are active. So similar to RAC, what happens if we globally activate rho A? Yeah, so the answer is clear probably. We will again have much less migration and a very stiff organization of stress fibers. But how do we activate rho A globally? There is a very simple trick actually to do that without having mutant overexpression. One guanine nucleotide exchange factor that activates rho A, which is GEF-H1, is associated with microtubules, and only when they are intact, when microtubules are intact, this GEF is inactive, right? When they are perturbed or they are dissolved, this GEF is released from microtubules and is activated by that, so it can downstream activate rho A. So this is something we did, or we often do. Actually, it's a very simple assay and very well known. So here the activator is released, and how do we depolymerize microtubules? By adding nocodazole to the solution. This is an actin depolymerizing drug. So, and the result is something like this. Sorry. Microtubules, what did I say? Yeah. Okay, microtubules, of course. Yeah. So, so the result is this, that we have really many stress fibers and almost no protrusions in those cells. But the cell again cannot migrate, and we have the same conclusion as with overexpression of a protrusion protein, RAC. Global activity does not, global activation of those GTPases cannot be the key factor for efficient or sufficient cell migration or migration efficiency. So it has to be a local regulatory mechanism, or maybe multiple regulatory mechanisms that control the local spatial and temporal activity, which is important for efficient cell migration.
But you are saying both molecules are expressed globally as well.
Say again?
You are saying both the RAC and the rho are globally expressed.
But they are not globally active.
Yes.
Yeah.
But they are. It's a yeah.
So in let's say in non-perturbed situations, when you have your cells that are migrating, you won't have active RAC everywhere in the cell. You will have it at certain spots, which I'll explain in a bit. Yeah. So and this location, wherever they are active, they can be basically effective also. Yeah. So this is the idea behind these many slides that I show. And in addition to those upstream regulators, GAFs and GAPs and others, it is basically generally thought that the GTPases themselves control the activity of each other, yeah, in such way that RAC and CDC42 stimulate each other and are more effective or doing basically their activation of effectors in the front, and that both of them actually act antagonistic to Rho A, that which then produces those gradients in the cell. Well, in order to understand or to study if those models are valid and how the localized signaling is really controlled, or what is the localized activation of GTPases is, more and more groups have studied or used binding domains of downstream effectors. Here, for example, CDC42 of CDC42 to study or to generate activity probes. Yeah, and the way how we use those domains is basically that we make a construct or produce a protein of this effector domain, and this domain will not interact with the inactive Rho GTPase, but it will interact with the active Rho GTPase. This is the basic principle. But what we need also is a signal, a reporter signal that is sensitive to this change, to this interaction, right? And one such signal, or a very much used reporter signal, is the so-called first resonance energy transfer, which describes basically the non-radiative energy transfer between two fluorophores. And basically several factors have to be fulfilled. For example, the two fluorophores have to have a significant spectral overlap, and they have to be in close proximity and orientation to each other. So what happens during FRET? So when we excite one fluorophore, such as for example CFP, the excited electrons fall back to the ground state and emit fluorescence, just as we see normally emit fluorescent light. Yeah. This is if no second acceptor protein is there. So if there is an acceptor protein with a proper spectral overlap is in the close proximity, this energy can be transferred by so-called dipole-dipole interactions, Wechselwirkung, which then leads to the emission of the fluorescence from the donor. From the acceptor, protein. So this is FRET. Yeah. How can we use this? Yeah. A colleague of mine, Olivier Pert, has used this phenomenon basically to when he generated this activity sensor for Rho A, where he had a single chain probe, single chain construct that encoded for the GTPase itself, then the two FRET pair fluorophores, and the effector domain that would only bind to Rho A. In this case here, we don't have any binding between this Rho binding domain and Rho, because Rho is inactive. Yeah? So the two fluorophores are not very close to each other. We have just little FRET energy transfer. Once the Rho A is activated, this RBD binds the active Rho, brings the two fluorophores together, and now we have very high FRET efficiency. So Olivier could very nicely show in migrating cells that Rho A is active in the front region, and that's persistently. So this was very new because, as I said, everybody was thinking because Rho A makes lots of contractility in the back, it must be highly active in the back. So this was quite new. But we could confirm this in other cell types as well, such as in HeLa cells here. And they are, when we did some movies, short movies also, and looked for the activity distribution using chymographs here, that the activity was highest in the very edge here, the very edge region of the cell protrusion. So this was again basically a confirmation that dynamic protrusions have activated Rho A elevated. Not only just during migration, also during the transition of those epithelial cells into migratory behavior, such as I showed before with hepatocyte growth factor, we found a really distinct difference in the patterns of Rho activity, whereas here in the epithelial shape, Rho A was barely localized to certain regions. In the migratory behavior, during migratory behavior, we have really highly activated, the GTPA is highly activated in the very, very edge of the proteins. So what does it do there? Well, before I come to the question why Rho is activated in the leading edge, I would like to show another probe that we generated for now CDC42, because this is another type of sensor signal that you can use basically for sensing interaction changes in the interaction capacity of proteins. So this method is based on the so-called solvent-sensitive myrocyanin dyes. The solvent sensitivity I would like to explain now. So those dyes are barely fluorescent in hydrophilic environment, basically because of the separation of charge. Yeah. So and if you put them in hydrophobic solvents like butanol, you have a very strong increase in fluorescence, which is explained basically by the increased delocalization of those proteins. So and that then it facilitates the fluorescence. So this is very much chemistry. I haven't done those dyes, right? I haven't cooked them. Alexei in our lab, when I was in Klaus Hahn's lab, did generate those dyes, and I put them, or I coupled those dyes with this domain, with a specific domain, effector domain, that will only bind to active CDC42, active GTPase. So what happened is this in vitro with purified proteins, that when we had no CDC42 active, this fluorescent signal was basically the same as if there was no CDC42 at all. So, and if you had active CDC42 together with this probe, then the signal would increase like twofold, almost. Well, when we looked at the structure, this was published, I forgot, sorry, to put the reference here. We found that the dye position where we coupled the dye with the binding domain would actually come into a hydrophobic pocket only upon binding between the two proteins. So this was very nice, because it would fit the idea that the domain, if it's just in water or in the water, it wouldn't have much of fluorescence, but only upon binding it would change its whole environment by being more hydrophobic. So, and in cells we had to have another control or ratiometric application, because in cells you really don't know just by the intensity of the light if there is more active CDC42, because it could be the effect of accumulation or thickness, etc. So you need a reference fluorophore basically, which is not sensitive to this interaction or to activity of CDC42. So we coupled this with GFP and did ratiometric imaging, basically divide the dye image by the GFP image, and then this is the result. What you get, that CDC42 is also active in those protrusions a lot, but it is also inside the cell highly active, and when we stained for with markers, we found that it's active in the trans-Golgi region, which is important for basically the polarity of protein transport during migration. So in summary here, basically we find, or we can measure activities of those 3 GTPases, yeah, but we find that all of them are localized, the activity is localized in the leading edge or in dynamic protrusions. So in addition to that, we looked for the colleagues of ours looked for the cross correlation of these three proteins activity signals with respect to the protrusion dynamics. The protrusion dynamics is here or starts here, and we see that rho A actually is almost simultaneously appearing with new protrusions. So which is again was very striking. What this GTPase actually could do in this very, very edge of the cell. So, yes.
What is plotted there?
Sorry? Oh yeah, okay. So this is the time, basically the time lag of, these are the signals, cross-correlation signals of each three GTPase sensor signals. They are cross-correlated to the protrusion dynamics, basically.
At the same location.
At the same location, at the very edge of the cell. So if so, you have Mahajek. I mean, he could really plot the cell, how the cell edge would translocate. Yeah, he would have the spatio-temporal data of this pixel based, and he could plot in this very region also the activity of each GTPase, right? And then you can cross-correlate that and look how this the maxima basically are shifted. You can read it really the way you see. If you have a protrusion initiating just briefly after that, rho A is highly active, then we have CDC42, and in a broader gradient we have RAC activated.
Yeah, what I wonder is what are the lines and what are the dots?
Oh, okay, I'm sorry. The dots are basically 95% confidence.
Ah, okay.
Yeah, so been a long time. So it is quite still it's very striking, right? So what does this protein do in the leading edge? So to study this, we now in my new lab, or not, in 2008 it was my new lab when I started. So we studied the relevance of those signals and the effects of those signals in a cell system which is very nicely suited to study cell contraction or cell stress fibers. Those are the sarcoma cells, U2OS cells, and they have a very, very nice actin or stress fiber organization with several different types of stress fibers. We have, I have some examples of here. We have those transversal arc-shaped, we call them arcs. Yeah, they are not attached to any substrate adhesion points. So no focal adhesion connection. Then we have those dorsal fibers which are attached to the focal adhesions with one end, and with the other end attached to those arcs. Then the third one are those so-called ventral stress fibers. With both ends they are attached to the substrate. So these three types of stress fibers have been shown to be also involved with tightly connected actually in their dynamics. So if we look at these are again the dorsal fibers. They are attached to those arcs, and the maturation happens in this way, that the arcs that are connected to the dorsal fibers basically fuse at some point further inside, like centripetally, after they moved, they fuse with those dorsal fibers with their flanking dorsal fibers, and to generate this elongated or straight ventral stress fiber. So this is the maturation process that is highly controlled in space and time. So how is this rho A linked maybe to this process? We know that, or we observe that those stress fibers or those arcs are generated from within this very lamellipodium. Yeah, we do see those flow of those arcs. And this is the first observation we have to keep in mind. So, and in those regions, what might happen? So Rho is known to activate those actin polymerizing formants, and they are known, or they have been shown to induce actin polymerization from those dorsal fibers, right? And we do know that Rho-A also activates this myosin light chain via Rho kinase by phosphorylation, so induces filament contractility. Yeah. A third protein that has been shown to be downstream of this ROCK is also phosphorylated, is the formine FHOT1. So it was very long not known what it exactly does, but it was shown already that expression of an activated FHOT would produce or generate those very pronounced stress fiber phenotypes. So this was known, but how this would happen was really not clear. So we found that, or we were able to show that FHOT, activated FHOT and myosin co-localize in the very edge of the cell, and those dots are actually organized within the transversal arcs. Yeah? So they are co-localizing in those arcs, whereas FHOT it does not co-localize with another component of those transversal arcs, which is alpha-actinin, that is important for the bundling of those structures, of those filaments. It is not co-localized. When we depleted FHOT, it did reduce the number and the number of those nice transversal arcs. This is basically, we measured the relative size of the lamellipodium or lamellum and the intensity of the actin in this lamellipodium or lamellum. Sorry. So, in contrast, when we overexpress the active form, we do see an enhanced generation of those stress fibers. This is a system to induce freshly actin polymerization. You destroy basically the cytoskeleton by using cytochalasin. This is a drug, a poison basically for the cell. Then you wash it out and you basically synchronized all the entire machinery to start the fresh actin polymerization. So we see that FHOT initiates or induces those arcs. Yeah? So those were the transversal arcs. They are positively regulated by FHOT. On the other hand, those dorsal fibers, yeah, they are attached to focal adhesions, they are negatively regulated by FHOT. Yeah? FHOT inhibits the growth of those dorsal fibers. So we measured it after overexpressing a dominant activated form again. We could measure basically the length of those fibers. It was significantly reduced. And also in FHOT depleted cells, which don't have functional FHOT anymore, the size of those fibers all of a sudden were enhanced or enlarged as compared to control cells. So this means that it negatively regulates those stress fibers, those fibers. So to understand this, or to have an idea why we have those opposing effects actually of this FHOT to different stress fiber types, I would like to show this data which Schönichen et al., our collaborators, published a couple years ago. So there they found out that FHOT, when you put FHOT in the same in the solution in vitro and measure the actin polymerization, this is this pyrene fluorescence is representing the amount of polymerized actin. Yeah? And this is another formine that is known to polymerize actin filaments, and you see that it really increases the pyrene fluorescence, whereas FHOT, full length or activated form, doesn't matter, inhibits even actin polymerization. This is actin alone. Yeah? So it does in vitro inhibit actin polymerization, similar to capping proteins. Capping proteins actually bind to those barbed ends of actin filaments, stop the elongation. Yeah? So, and this capping role, capping function would basically explain both observations we had. Yeah? So it would explain why arcs are enhanced, which are basically generated by those filaments in the leading edge, Effort would, by capping, enrich those small filaments. It would prevent filament elongation, but enriching the small filaments. This will give myosin and other regulators the chance to organize those small filaments much better. And then the arcs would be able to fuse basically with the dorsal fibers, which are represented here. And in those dorsal fibers are organized by parallel and long fibers, and that's the key. They do require elongation for their shape, yeah, for their size. And if you have effort in the cell, it will prevent then, by its capping mechanism, the elongation, and you end up having shorter dorsal fibers. So overall, we think that the balance activity of F-hot is important to both organize the length of dorsal fibers and the generation of arcs, so that they can find each other properly and generate the final mature ventral stress fibers. So that's basically the last slide. I think I have the shortest talk today. So this is the last slide where I try to kind of explain why this activity of row A in the front makes now perfectly sense, right? Because many of the processes that are required for generation and maturation of stress fibers happen here, yeah? So it is on the one side that the dorsal fibers are initiated from focal adhesions here. Then on the other side, the arcs are generated from within the lamellipodium. You all here need basically the downstream effectors to be active in the very front as well. So for that you need, it is perfectly clear that rho must be in the front, yeah, activated. So these are the my people that so former members. Nina Schulze did a lot of work with the forming with F-hot. Vera Schwarz worked a lot with sensors, with the Fred sensor, and HGF stimulation of epithelial to mesenchymal cell migration. And Melanie and Johannes are still working on F-hot projects on different aspects. And collaboration, I put here also my former supervisors and later collaborators, Klaus Hahn. He does a lot with the work with biosensors. Gary Bockhorst, I went afterwards for my second postdoc, so to be more involved in the biology of those GTPases, and not only being able to generate and assess sensors, right? So altogether, Lou Hodgson and Alexei Tuchkin have helped a lot in those groups. We work together. Current collaborators, Leif Demelt from Max Planck Institute in Dortmund, and Matthias Geier, more on the with Leif we work a lot on turf microscopy and formin biology. And with Matthias, we did quite a lot of interactions on biochemistry, on the biochemistry of formins. Yeah. So, and yeah, thank you very much. That's it.
Questions? I have a question regarding these biosensors.
Yeah.
Now, basically, it's always a hairpin that opens and closes. And in one case, you have a specific interaction. Now, if you have anything that would compete for that interaction, you would get a false negative. You could have the closing prevented, so you would have a wrong reading. And in the other case, the primary sensor is basically the immediate environment of that dye. So any molecule that would bind in the vicinity of that dye would give you a change in intensity.
Okay.
And that, I can imagine, could be quite unspecific. Can you comment on these effects?
But you can do your proper controls, right? So you would have.
As always.
Yeah. So so the first one I thought I think you mean the Fred sensor, which opens and closes. because the domain is in closer proximity to the sensor, to the GTPAs, it is basically favored as an interaction partner with respect to other proteins that might be flying around or swimming around. Yeah, so it is indeed favored.
Experimentally proved? Because you also you bend the hairpin.
You mean the interaction with the hairpin itself? No, I mean it's not a hairpin. We have a linker, and Olivier tested like several lengths, several modules, which gave him basically the best response. You know, the way I think the best way you test those sensors is doing in vitro like titration curves and exactly knowing how much protein you have and basically titrating that. And doing this with another probe that wouldn't give you the this would have the same structure, but maybe with a mutation that would not bind Rho, yeah, even if it was active. And I mean, this looks quite nice. At least with the dye sensor, we did a lot of such work. Yeah, so we can say though that CDC42, you're right, is not the only protein that it binds when it's active, but it is there are two very close members of CDC42. The domain binds them as well, but with much less efficiency. You can measure this in vitro as well with a titration curve. Yeah, you have to do those things. That's why sensors are not very easy to generate. At least how to say, to be sure that it's really working, or if you're not, so that you're not measuring some false positive signals. I mean, the false negative signals I think are not very critical. I think the false positive signals are much more of a problem. Yeah, that people I think would publish quickly just because they do see some elevated signals in their ratio images, for example. Yeah, you have to think always. Although it takes then longer to finish the studies, obviously, but I mean we get more and more experience, and then with each project then has maybe less needs less time. Yeah, because we get more experience with each biosensor. Yeah. Yes.
Yeah. Maybe I have a question a bit genuine and not maybe so closely related to the talk, but related to the pathways that you described. So there are, basically for mammalian cells, two drugs that are often in use to inhibit contractility, which are blebistatin and C3. And I think they do not act exactly at the same level.
No, they don't.
And sometimes, so what is your opinion on?
C3 inhibits Rho upstream, and blebistatin inhibits myosin, which is downstream. Yes.
Yeah. Mm-hmm. So so when we use these drugs, sometimes like the macroscopic effects are quite different. Like for instance, for cells in channels, like the effect of C3 is to accelerate the front, while the effect of blebistatin is to decrease the front velocity. And
velocity of motility?
Of the moving front of the whole tissue.
Decrease, okay.
And do you have some kind of explanation maybe of why this is so, and is it like, I mean, in a sense, my question is it clear what is the effect of C3 macroscopically, and what is the effect of blebistatin, meaning like for instance, C3 inhibits like connectivity of the network, and blebistatin more something which has to do with the stall force of the contraction. Is there some kind of I don't know, it's a bit out of the blue, but do you have an opinion on this?
Let me try to answer out of the blue. So I mean, myosin is important actually. So is such key to this process of this physical process of motility, right? So if you inhibit this, I think this is not surprising that if you inhibit myosin, that the cell doesn't migrate anymore. That part is not surprising. right? What I can imagine, what is surprising is, or might be surprising, is that C3 or inhibition of Rho enhances now migration. But you have to think of that C3 also inhibits Rho B and C as well, right? So there are different. They are also. I don't quite remember, but one of them is also important for motility. Yeah. So I'm not sure if it works if you inhibit all three Rho A family members and then you expect one effect, right? So it is probably much more complex what you're doing to the cell if you add C3. C3 is actually usually used just to, you have your RNAi experiments or mutant experiments, and then at least I do that. I use C3 just as an additional factor to inhibit Rho A, or also Rho A. Yeah. So I mean, it is as the only factor to perturb, to use for perturbation. I don't think that it is working. It is working because it is much more complex with this C3 what you're doing to the cell. I'm not sure if it's sufficient, but I mean I can't really answer that question. Actually, I don't really know what happened. I mean, you have to also not, of course, an additional level is the effector level. Even if Rho was the only Rho GTPase that is affected by C3. You will have probably opposing effects also, just because that then kind of equal each other, maybe, because the effectors do so completely different things. Yeah. Normally, if you have local spatiotemporal control of Rho, you have maybe in some location or region effector X activated, and in another region effector epsilon activated. So you kind of control also their activity by spatial space and time. Yeah. But if you globally knock down Rho A, I know that RNAi treatment just makes a rounded cell, and the cell doesn't do anything. I mean, it is not only the global activation is a problem; it's also the global inactivation that it's a problem. So this is again, maybe I repeat myself, but C3 it's a critical experiment to do because too difficult to interpret.
Right.
Okay.
Any further questions? If not, we finish.